Okay, let's talk very quickly about the Calvin cycle. So let's start off with some fundamentals. Calvin cycle occurs in the chloroplast, so let's kind of start off by drawing a chloroplast. Doesn't have to be fantastic artwork, just good enough. So here we've got two membranes, and so the outer membrane is obviously called the outer membrane, the inner membrane is called the inner membrane, and then we're going to have some stacks of membranes. Let's draw those in. Okay, so the first question is, these membranes have got a special name. What are those membranes called? Okay, so what is a stack of those membranes called? Okay, so on these membranes, on these thylakoid membranes, that's where the light reactions are going to occur. So I'm just going to abbreviate that as LR, the light reactions. And so the light reactions are going to take water, and they are going to use the water and they'll produce some oxygen, and we will take the electrons from the water, and the light reactions will energize those electrons and eventually give them to a molecule NADP to produce NADPH, and the light reactions will also produce another molecule. So what is that other molecule produced by the light reactions called? Okay, so we've got NADPH and ATP being produced by the light reactions, and they are going to be fed into the other main set of reactions that we see in photosynthesis, and that's the Calvin cycle. So let's just represent that with a circle. So if we feed in ATP and NADPH, we're going to use the energy stored in the bonds of the ATP, and we're going to take the high energy electrons from NADPH, and so out of Calvin cycle is going to come ADP and another molecule. And so what is the other molecule that comes out? Okay, so we've got ADP and NADP plus that come out of the Calvin cycle, and they obviously get fed back into the light reactions. So that's the kind of relationship between the light reactions and the Calvin cycle. Now let's look at the main functions of the Calvin cycle. So the Calvin cycle is going to take carbon in an oxidized form, and so carbon in the form of carbon dioxide is going to come in from the environment, and then there'll be a series of chemical reactions we'll talk about in more detail in a moment, and those chemical reactions are going to be responsible ultimately for producing sugars. Okay, so that's kind of the big picture, and let's break it down and now look at it in a little bit more detail. So let's take a look at the next slide. Okay, so here is the diagram that I just drew, obviously a little bit nicer than how I drew it just now. But let's take a look at the details of the Calvin cycle. So we've got to keep track of energy and matter as always. So the matter that we're really going to start looking at is the carbon dioxide that's coming in from the environment, and we're then also going to track the ATP and the NADPH. We'll come back to those in a moment. So as what we're going to do is we're going to keep track of the carbons here. So let's start off with the first set of reactions in the Calvin cycle, and those reactions are going to occur here, and they are called carbon fixation. And so carbon fixation takes carbon in carbon dioxide form, so in a gas, and takes the carbon and attaches it to a solid molecule, and so that's why it's called fixation. So let's start off with that solid molecule, and let's do 6 of these at a time. Okay, we're going to do 6 carbon dioxides at a time and 6 ribulose bisphosphates at a time. It's easy to think about 6 carbon dioxides at a time. Okay, so we're going to take 1 carbon dioxide and add it to 1 ribulose bisphosphate. And each of these ribulose bisphosphates has 5 carbons. So there's the 5 carbon ribulose bisphosphate, and we're going to add 1 carbon from a carbon dioxide to the 5 carbon ribulose bisphosphate. Okay, so that's going to give us a 6 carbon compound, because the 5 carbons in the RUBP plus the 1 carbon from carbon dioxide is 6. 5 plus 1 is 6. It's pretty straightforward. Okay, so we're going to do that another 5 times. So what we're going to do is take a total of 6 ribulose bisphosphate. We're going to add 6 carbons, 1 each from a carbon dioxide to each of the ribulose bisphosphate. And that's going to give us 6 carbon compounds, and they're very unstable, and they immediately break down into this molecule here, phosphoglycerate. And so that molecule has got 3 carbons. And so because we've got to maintain the matter and we've not taken any carbon out, there must be 12 of them. So we went from 6 5-carbon compounds. We added a carbon to each of those. That gives us 6 6-carbon compounds. And then we're going to split those 6 6-carbon compounds into 12 3-carbon 3-phosphoglycerates. That whole set of reactions is called carbon fixation. The second set of reactions is called reduction and sugar production. Okay, and that's the set of reactions at the bottom here. So we'll come back to looking at the ATP and the NADPH in a moment. All we're going to say here is that the 12 3-phosphoglycerates are going to get converted to a molecule called glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate's familiar, and you may have seen this if you looked closely at glycolysis. When you split glucose, you eventually produce the molecule glyceraldehyde 3-phosphate, and it's converted into pyruvate eventually. So we're going to have 12 of those because we had 12 3-phosphoglycerates, and so we're going to take out two 3-carbon G3Ps, and so that's going to give us the ability to make 1 glucose, and that glucose can go on and be used to form starch or cellulose. It can be converted into nucleotides, into other sugars, all kinds of things. So that is a process of reduction and carbohydrate reduction. We'll come back and look at the reduction piece in a moment. So what we're left with is a certain number of glyceraldehyde 3-phosphates. So we're going to take those 10 G3Ps, and in our next stage, which is called regeneration of RUBP, which is this set of reactions here, is what we're going to do is we're going to reshuffle the carbon in the 10 G3Ps to go back to starting with 6 5-carbon ribulose bisphosphates. So we've got 10 3-carbon G3Ps, and we're going to end up with 6 5-carbon ribulose bisphosphates. And so that starts the cycle again, and then we can put in another 6 carbon dioxides and do everything over again. Okay, let's go back to looking at the energy usage here. So we're going to use some energy in the form of ATP when we do the reduction and carbohydrate production stages of the Calvin cycle. So that's going to require 12 ATP, and then we're going to use another 6 over here on the left when we do the regeneration of RUBP. So this cycle, taken as a whole, is highly endergonic. It requires a large input of energy in the form of chemical potential energy in the form of ATP. And let's look at this NADPH to finish up here. And so that NADPH, remember, is a source of high energy electrons. And so we're going to use that in the reduction phase. We're going to take the electrons from NADPH, give them to bisphosphoglycerate, and that's going to produce our glyceraldehyde 3-phosphate. So that's the Calvin cycle. Last thing to note here is that you need 50% more ATP than you need NADPH. And so that should get you thinking about what happens in the light reactions of the reactions of photosynthesis. So that's it. That's a quick walkthrough of the Calvin cycle.